Morning students. Today in 10th standard we are again going to continue with the chapter on photosynthesis. And you know that in the last class we had ended about the starch test, which is used as a determinant for testing any experiment where photosynthesis has taken place or not. So you remember, I hope you remember that what is the method, that how we test for starch. Starch is tested with iodine solution, and wherever starch is formed, iodine gives blue-black color, or where starch is not formed, it gives brown color on a decolorized leaf. Now, today we are going to study about the experiment on how to show that chlorophyll is necessary for photosynthesis. Chlorophyll, it's one of the organic ingredient where magnesium is one of the main element found in that chlorophyll molecule, and chlorophyll helps in trapping the sunlight, and it is one of the most important factors. So how chlorophyll is required for photosynthesis or not is determined by an experiment where we take plants like coleus or crotons. Coleus and crotons have variegated leaf. Now variegated leaf means if a leaf has different color. You can see here I have shown a croton leaf or a coleus leaf. This is the margin of the leaf and this is the leaf blade, and here you can see it is colored green and rest of the place it is white. So you will see that this type of variegated leaves are found in various types of show plants which are generally called croton. So you have to take a discharged plant, a croton or variegated plant. You know how to discharge it, keeping it in absence of sunlight for 2 to 3 days, and then you expose the whole plant, potted plant, in sunlight, and you pick up one such leaf where you have a variegated leaf where you have green color inside or white color outside like this, or it can be of different shape. Now you have to draw a diagram and mark the color of the different parts of the leaf. Now that leaf is plucked and then it is usually sent to the same procedure. We dip it in hot water to stop the metabolic process, then boil it in alcohol in a water bath to decolorize the chlorophyll molecule, and then the leaf becomes off-white. We take out the leaf becomes brittle, as because when you boil in alcohol, usually it is the water is absorbed, or what is called the alcohol takes out the water, the leaf becomes brittle. So we dip it in warm water and spread it on a petri dish. Now when we put iodine color, you can see here wherever the green color was there in the leaf, in that part the blue-black color is formed, showing that starch is formed. Rest of the places where white color of the leaf was there, it shows the brown color of the iodine along with the decolorized leaf color. So this is actually showing that chlorophyll is necessary. So wherever chlorophyll is present, photosynthesis takes place. Where chlorophyll is not present, photosynthesis does not take place. You must know the example of these two plants. The two plants are croton, Croton, and coleus, Coleus. Sometimes they are asked that name to plant which has variegated leaf. Now the next experiment is to show that oxygen is given out during photosynthesis. Usually, this experiment we use in various other places to show the rate of photosynthesis, how it increases, like say for example with the increase in the CO2 concentration, or with the increase in say the light intensity. We use the same experiment because in this experiment the oxygen that is liberated, that is directly proportional to the factors. That means if you increase the CO2 concentration, the oxygen bubble that comes out from this experiment is more. Anyway, that is a different issue. We are going to study about this experiment, and where we show that oxygen is liberated. What are the requirements? We require a say 500 ml beaker. So this is the 500 ml beaker. We need a short stem funnel. Short stem funnel is a funnel the stem of the funnel is broken, because this stem should not cross the level of the water here. So that's a short stem funnel. We need some aquatic plants or weeds like hydrilla. You must know the name, Hydrill. Hydrilla plants and there is a support of the funnel here at the bottom. We need a test tube which is considerably little bit big. Now what we do, first we take water in the 3/4 of the beaker filled with water, and then we put 2 or 3 pieces of hydrilla plants. Now these hydrilla plants should be freshly cut hydrilla plants, and it should be kept in water beforehand. When we cut this plant, we cut with a sharp instrument, because the stem of the funnel should be so sharp that it should liberate the oxygen. So we take 2-3 sprigs of this hydrilla plant, put it inside the funnel, and place the funnel inside this beaker containing water. A test tube which is taken, a test tube is filled with water and slowly inserted or inverted over this funnel in the beaker. So, and when you fill the invert the test tube, the water level in the test tube is till the top. Why? Because the atmospheric pressure pushes the water till the top. So there will be no space almost at the top. Now, if you keep this whole setup in sunlight, then what you will observe, that slowly you will see bubbles of gases coming out from this and accumulating at the tip of the test tube, and slowly the water will be displaced downward. So there will be a gas collected here. Now this gas, we don't know what gas is this, but if we test this gas with a glowing splinter, when this test tube is full, we take out the gas, take out the test tube and insert a glowing splinter. We see the splinter burst out in flame, showing that it is nothing but oxygen. Or you can test it with alkaline pyrogalol, which turns brown. So this is an experiment to show that oxygen is liberated during photosynthesis. You know that oxygen is one of the byproduct here. So you can see that this question has come several times, putting up this experiment. So this is a test tube, the oxygen, the leveling you must know, the short stem funnel, hydrilla, and this is the support. The support is given so that the water circulates inside the funnel also. Now this experiment, you must sometimes they give this diagram, and you must remember that when you draw this diagram, it should be exact. The stem of the funnel should not come above the water. These are the precautions. The hydrilla plant should be sharply cut, and before inserting here, it should be kept in water. So these are some of the precautions that we see. The next experiment is now this is the factors affecting photosynthesis. Now there are several factors which affect photosynthesis. Some factors are external factors, some factors are internal factors. Now the external factors are like, so for example, intensity of the light. Now if the intensity of the light increases, the rate of photosynthesis increases proportionately, because but to a certain limit, because too much of intensity of light may kill that stomatal apparatus or destroy the stomatal apparatus due to solarization, and then it may reduce. The second is that the temperature, the rate of photosynthesis with temperature. The temperature also, if it increases to a certain limit, the photosynthesis increases. The right temperature is about 35 degrees centigrade is optimum temperature. It should not be too high and it should not be too low. So the optimum temperature is needed, because most of the reactions of photosynthesis are enzyme based reactions, and the enzymes get denatured at high temperature, and also the work of the enzyme may not be so efficient when the temperature is too low. The third thing is that the CO2 concentration. Now CO2 concentration is also an external factor and internal factor also. Like CO2 concentration, if you increase the CO2 concentration, the rate of photosynthesis will increase, and if you decrease it, it will decrease. But in CO2 concentration, if it increases inside the cell, sometimes it induces the stomatal closure. So that may cause reduction in photosynthesis. Water content also, if the water content is less, the rate of photosynthesis also decreases. Similarly, in the internal factor, chlorophyll is one of the factors. If chlorophyll is not there, then photosynthesis will be less. The structure of the leaf, if the leaf is of a different structure, if the layers of the palisade layers are less, then rate of photosynthesis may be less. The shape of the leaf, the arrangement, the phyllotaxy of the leaf also greatly affects the photosynthesis. Now, internal factors of CO2, as I already discussed about, and age of the leaf. As the age of the leaf increases, usually the chlorophyll is taken out from the leaf, and then the rate of photosynthesis naturally decreases because the chlorophyll is less in an agent. That's why you see leaf becomes yellow or decolorized when the leaf ages. Now there is a law called Blackman's law of limiting factors. If you make a graph here, you can see here in the y-axis is the rate showing the rate of photosynthesis, and in the x-axis it is showing the light intensity. So it says that the factor which is the slowest factor, if there is an increase in the slowest factor, then there may be a rise in the rate of photosynthesis. Now Blackman's law of limiting factor says that suppose this is the CO2 concentration, the rate of photosynthesis is this is the graph showing the rate of photosynthesis, and it becomes optimum after some time, and then it becomes straight, that is 0.03% of CO2 at a temperature of 30 degrees centigrade. Now at this particular point, if you increase another factor, say for example you increase the intensity of the light, then there may be a further increase in the rate of photosynthesis. Or if you increase the CO2 concentration with the same light intensity, there will be still increase in the rate of photosynthesis, but it will become limiting after some time. So this is Blackman's law of limiting factors. Of course, this is not in details, you don't have to study, but you say that when the slowest factor, if it is increased due to some other factors, then there is a slight rise in the rate of photosynthesis, but that also becomes limiting after some time. So these are mostly the things that we study here in photosynthesis. We have to know about that how does photosynthesis help us. Like photosynthesis help us directly by formation of the food, or we get oxygen from photosynthesis, which is keeping the balance of oxygen in the air. Photosynthesis indirectly serve food for other animals also, because it helps in preparing the plants to prepare the food, and the herbivorous animals are dependent on that, and the carnivorous animals are dependent on the herbivorous animals. So if you are asked that what is the significance of photosynthesis, you are right, it helps us getting the oxygen or balance of the oxygen, it helps us in procuring the food, it helps in procuring food for the animals of different kind. So this is all about it. And the carbon cycle, you may have a little bit knowledge about it. How does the carbon cycle throughout the world, like say for example how the CO2 is fixed by the plants, and that plants are converted to fossil fuels like coal and petroleum in the sea, and then again when they burn, the carbon dioxide returns to the air, and one form of carbon is the fixation of carbon into the plant body, forming carbohydrates and proteins of the body. So like this cycle that is going on in the environment, the balance of this carbon is called the carbon cycle. So thank you very much. We'll go to the next lesson in the coming class, and keep reading the chapter. If you have any question, you can put up the question. And once again, thank you. Keep safe from the virus, and all the best.